The issue with like big molecules, I mentioned early on, antisense oligonucleotide, sRNAs, and zinc fingers. These are things that are need to be delivered into the cell or into the brain. And predominantly, Huntington's, everybody thinks of it as a brain disease, and it is predominantly the cells most sensitive are cells in the striatum, but it is all expressed all over your body. And there are other components that happen, including your motor neurons, which is why initially Huntington's was called Huntington's chorea, and there's this dance-like movement, and they twitch and they move and etc. So there's a component in the peripheral that actually is impacted, but the most devastating part is in the brain. And the question is, how do you get it there? Because, a small molecule, you take a pill, they get absorbed, and it crosses the blood-brain barrier. Your brain is nicely protected for obvious reasons, right? Your brain is protected so no harmful components can come, so you can still think, because it's the computer that orchestrates everything in your body. So in order to deliver those therapeutics, which let's say they're called large molecules, you need to actually drill or actually put it into your through your synovial fluid. So those are challenging things to do. And what we are doing at CHCI is looking at different technologies and delivery systems that will avoid to use sort of a drill bit, avoid the drill, and sort of we're looking into actually encapsulating viruses that do not have any harm, where you can actually inject it IV, or at least, a simple injection as opposed to a drill intracranially. And we're looking at other type of delivery systems. We can use any of the transporters that are available within the brain to try to get that carrier. They call leader sequence, and sort of Roche calls their technology the shuttle, right? And Roche also bought the ISIS ASOs for Huntington's because of us. We spend quite a bit of money, not only money, but also scientific and strategy and input in catalyzing ISIS to actually develop an antisense oligonucleotide for Huntington's. That particular molecule is not what we call allele-specific. It means it hits both the mutant and the normal Huntington, but we believe that if just lowering it 50%, we could still get a major impact on the disease. But then again, that also has to be delivered, and that's the challenge with those technologies. But obviously, we believe that we need to do what we can to solve this problem. So we're basically going after every possible technology that exists out there to modulate that gene and that protein, and then are now understanding how to best deliver that without being an invasive drill sort of format.